all online. If there are any newbies in the crowd, I'm Peter Steinberg. I'm the director of the World Harbour Project. I want to welcome you to our fourth webinar. And I was going to do just a few words by way of introduction before I hand it over to Anthony and then Aina. However, while I've got you on the line, so to speak, before I actually begin the introduction for the webinar, I did just want to do one other piece of World Harbour Project business. And that is we've been discussing with a number of the partner cities the possible location of the next Global Partners Workshop. So in fact, we've only had one previously, the initial one at Sydney. So the plan is to have another one this year. Yan Wei has kindly agreed to host it in Xiamen. Originally, we had planned to do this in either May or June. I think that's now too soon. So it's most likely to be either late September or early October. So sometime in the next couple of weeks, we will send something out that firms up those plans. But I just thought I'd give you a heads up while we were doing the webinar. So that's Xiamen in late September, early October. And as I say, we'll send out more details in a little bit. Okay, so today in the webinar, we're in the space of Working Group 1, Water and Sediment Quality. And it's, as you know, one of the four thematic areas of the World Harbour Project. And there are two talks on two aspects of that theme that I think are both really quite important. So the first talk is from Anthony Chariton, formerly of a government research organization in Australia, the CSIRO, but who has recently decided that he's going to live the high life in academia and has moved to Maquarie University in Sydney. So congrats on the new position and good luck with that. So Anthony is going to talk about some of the modern molecular techniques that are increasingly being used for assessing the health of estuaries and other marine environments. And it's something that we intend to and have been trying to bring into the World Harbour Project in one context or another. And I think working with Anthony and others, we may finally have found the right project to do this in the context of the WHP. So Anthony is going to talk about some of the work that he's been doing with, as it's called, metabarcoding. The second talk is about pollution and contaminants. And that's by Aina Gomez from IH Cantabria in Santander, Spain. Now, one of the key issues for all our harbors are pollution and contaminants. But one of the things that I like about the approach that the folks in Santander, and Aina in particular, have been taking is that, you know, it's not just about the presence of the contaminants. It's about the environmental risk and the consequences of those contaminants. And so Aina and her colleagues have developed a number of approaches for trying to do those environmental risk assessments. And again, it's something we're right in the midst of talking about bringing into the World Harbour Project and applying it across a number of our different cities. And you will be hearing more from me on that over the coming weeks as we roll out some of those techniques. So it's about Working Group 1 tonight, or this morning, or whatever it is, wherever you are. And I look forward to hearing what first Anthony and Aina have to say. As has been the custom with these webinars, we'll go through both presentations and then we'll open it up for questions. So over to you, Anthony.
Oh, thank you, Peter. Well, good day, everyone, and good morning and evening to everyone around there. So this afternoon, I just want to talk about, there you go. So we'll talk about using metabarcoding to look at benthic communities. So I want to give an overview of what's sort of driven this research. And I'll talk about how it's actually done, give an overview, and then give one case study we did in Southeast Queensland, and finally talk about some of the issues and possibilities on the way forward to refine the approach. So just want to thank Peter and Karen and acknowledge my co-authors. So as most people are aware that The most common way for assessing estuarine health is to actually look at the macrobenthos, and so you're looking at things like polychaetes, bivalves, amphipods, etc. However, this process is very costly, so for us it costs about $300 US to get one sample processed. You need numerous replicates to capture the spatial and temporal variation in macrobenthos. And what that means is you've got hundreds of replicates, is that it often takes 6 to 12 months to produce the data. And this has been quite a challenge because in looking at impact work, I work with toxicologists and chemists, and they've often finished their work in 3 months, and about a year later I'm putting my hand up. And the project's moved on. One of the other challenges we have these days is finding local taxonomic expertise, which is getting harder because there seems to be less interest these days in people doing it. And we also found that many of the systems we work in, for example, deep sea and also high energy environments, there's actually not enough macrofauna to make any statistical comparisons about. So you only might get 1 or 2 organisms per sample. And for me, one of the biggest challenges is that I find the approach has a limited sensitivity when detecting change, and this especially a modified system. So you can tell the difference between a very good system, a pristine, and a very poor system. But if you're looking at moderate changes, based on my experience, is that macrofauna have not been sensitive enough. So, as you can see here, it's really this area of uncertainty in the middle that it throws the challenges, and especially if you're working in harbour, so you're already working in systems that are disturbed. So this is the area of the greatest challenge. Can we increase the sensitivity in this middle area here? And for me, one of the biggest issues of macrobenthic surveys is that what we're doing, we're looking at about 50 or 60 taxa, and we're making assumptions on total ecosystem health, looking at the trajectory of this small number of species. And you can see here, it's just the tree of life. This is just for those species that have been identified. So it's obviously got a lot less bacteria, fungi, and archaea than normally go. But you can see we're only looking at a couple metazoans, and we're just looking at those and we're making assumptions about ecosystem health. So really what we're trying to do is there a way to get a more holistic view of biodiversity, get a cheaper and faster and a timely approach so we can start really providing a new way of looking at ecological impacts within disturbed systems. So for those who are unfamiliar with the approach is what we call metabarcoding. It's amplicon sequencing is the common name. So the idea here is we go out and collect samples. In this case, we've got sediments. We extract the DNA from the samples and purify it. And what we do is we amplify particular genes of interest. For example, in this case, today's talk, we're going to talk about a gene called 18S RDNA, which is found in all eukaryotes. And you can tell if it's a polychaete, it's more similar to another worm, and it's less similar to a fish. So you can use those genes, but you can use a whole suite of genes to get different types of variation in bacteria and different taxonomic groups. And what basically, you get the DNA and we amplify it using PCR, so we have a pool of products. And this itself is quite important because what this does is pose a challenge because PCR has a lot of issues associated with it. So if you've got a complex mixture, the chemistry might like dogs more than cats. So it over-amplifies dogs and under-amplifies cats. So what that means is that the proportional abundance and the true abundance of your sample is skewed. So often, as in this demonstration I'm going to do, I'm just going to talk about presence-absence data. So once you've got all your samples, you can get hundreds of samples together with lots of different genes. You can do what we call multiplexing, where each sample, we put a little sequence on the front of it, ATGGC or GGACC, and that means that that's from sample 1, replicate 1, or sample 4, replicate 8. And what we can do is we can put hundreds of samples together, run on a machine like this HiSeq 2500 and sequence them. And then the sequences come out, and then we know that this sequence belongs to treatment 1, rep 1, etc. So you can process hundreds of samples in one, so it's good for experimental design. So once you get the sequence data out, you just get this kind of GATC stuff, and that basically needs to be cleaned. There's lots of errors, and once we get cleaned up data, we can hit it to a taxonomic database, such as NCBI or Silver, and we can provide some taxonomic assignment to those sequences. You can see the first hit we got from this sequence is Capitella capitata, the polychaete. And then we can assign some taxonomy. And it's important to realise here that if you want really good taxonomy, you really need to build your own database. So if you're interested in your particular benthos you've already been working with, it's best to build a sequence database out of those, and then you can get far more accurate and more confidence with your taxonomic resolution. So another really important thing to think about is what we're doing using this approach is that we're just taking a whole sample of the core. So in addition to those macro benthos you would normally sample, such as your bivalves, polychaetes, etc., we're also picking up things like larvae, biofilm, small cryptic taxa, non-metazoans like diatoms. You can also pick up parasites and eggs and cysts and dormant things, the gut contents, deceased organisms, and catchment vegetation. So this is quite important. So it gives you a very different view of biodiversity. There are ways around to deal with some of these things, like looking at RNA to remove the dead fauna or something, and also filtering and looking at different sized genes. But for us, we're trying to do a rapid approach, and this is the approach we've undertaken. So I just want to talk you through one quick example that we did, which is up in an area of in Australia called Queensland, Southeast Queensland, which is up here. It's probably similar to Florida, would be the most common thing. And basically what they do is the government every month goes out and measures 18 estuaries along the river here and 18 rivers. And at the end of the year, they get a scorecard. The rivers get rated from estuaries from A to F, A being excellent, obviously, and F being a fail. So they've got lots of estuaries to do, and they're doing it monthly. So the issue is that there's no way they can collect macrobenthics data to provide ecological endpoints. So to date, really, it's based on mainly on physical chemical measurements, such as nitrogen and phosphorus, turbidity, and also habitat. So what we want to do is to see if we can piggyback on top of this project and see if we can provide rapid ecological data. So the main things we're interested in is, can metabarcoding discriminate between the estuaries? Can it identify where the metabarcoding is correlated? Does it show strong correlations with environmental variables? And does it actually provide any useful ecological data? So in this study, we looked at 5 estuaries. That's the Noosa up the top here in the north, which is a common tourist place, the Maroochydore, which is the intermediate impacted sites, the Currumbin, which is also intermediate, and the Logan down here, which is an F. The Logan is a highly eutrophied system. So for today's talk, the main things to focus on is that the good ones, the Noosa, the really bad ones, the Logan, and the other ones are different, but they're all sort of intermediate. So just to give you a view of the estuaries here, this is the Noosa, which is the good one. You can see this touristy area down in the front here, but once you get upstream a bit, you get into this national park, which looks pretty, but it's actually sort of mosquito infested. And if you look at the Logan here, you can see that it's very turbid waters. It's got high eutrophication. There's agriculture, and a sewage treatment plant. So it's actually changed, as you can see, quite dramatically from the image above. So we went out and sampled these estuaries using a similar kind of approach you would do normal macrobenthos, running up the estuary, up the gradient, taking multiple replicates at each site. So here's just a real summary of the results we got. I just want to point out, we're talking a Metabarcoding and epicot sequence. The results are called OTUs. These are operational taxonomic units. They're unique gene sequences. That doesn't mean they're always a new species, because some species have multiple OTUs, but that's the molecular unit we're talking about. And I've just clumped them up here at phyla so you can get a bit, because there's so many taxa, but the most important thing you can notice is that we're covering a huge range of eukaryote taxa here, from the diatoms to nematodes to fungi to annelids, etc. And here's the 5 estuaries. So remember, the Noosa is the good one, the Logan's the bad, the other in between. And the most striking result you can see is that the biotic richness, the higher richness, biodiversity richness, is actually in the Logan, that's in the most polluted estuary. And this is because the Logan actually has far more catchment inputs than the other estuaries. So you've got these fish farms down here, prawn farms, you've got agriculture runoff coming in, and you've got a sewage treatment plant. So while the biodiversity might be higher, it doesn't mean it's desirable biodiversity. And we'll talk about that in a bit more detail in a second. And this is a common trend we find when we're doing these metabarcoding surveys. So if you look at the graph here, this is a traditional pattern you find with richness along estuaries. So in the ocean, you always get a high richness. It peters out in the estuarine area because of the physiological tolerances of organisms to deal with estuarine conditions, and then increases again once you get to freshwater. However, when you're doing sequencing, we always get a different result. We get these peaks in the middle, just like this, and every time we sample system. And again, this is because there's more tributaries coming in our systems in the middle of the system, and every time we hit a tributary, it fluxes up our biodiversity. So I just want to emphasize it's a very different measure of biodiversity than traditionally obtained. So we just want to look at composition. So remember, the first thing we want to do is, can metabarcoding discriminate between the 5 estuaries? And here's just a simple MDS, and you can see here, here's the sites from the Noosa, here's the Logan, and here's the other 3 estuaries. And the most important thing is we found that each estuary contained a unique composition. They were significantly different, but the most obvious thing here is that the most different systems are, in fact, the Noosa, the good one, and the Logan, the poor one. So, first tick of the box is, can you discriminate between the estuaries? And yes, you can. So the next thing I wanted to do is see and go, is there any particular taxa at the time of sampling which were indicative of the systems? And to do this, we used an R package called interspecies, which is indicated taxa. And basically, the software identified that there's about 400 potential indicator OTUs. And these are potential because you need to sample many times to actually get real indicator OTUs. So what we found was really that in the Noosa here, most of these belong to were bacillophytes belonging to Bacillophaces. And then in other systems had different taxa contributing. And most importantly, when you looked at the Logan, the poor one, a lot of the taxa were associated with the breakdown of organic materials such as these fungi like chytrids and ascomyda. So you can already get a feel that each system has a particular different biodiversity, which is unique to it, and some of them are more reflective of the environmental condition, of poor environmental condition. So the next thing we wanted to work out is there a strong, do you get a relationship between composition and the key environmental variables of the system? And for here, we've just got a basic a distance linear model, and you can see here these sites. I've just pulled them up at sites, the replicates just to aid. And you can see this is the Logan, and here's the other estuaries, and here's the environmental variables. And the most obvious striking thing here is that the Logan sites are more strongly correlated to things like turbidity, nitrogen, and phosphorus, while all the other estuaries, you can see here, there's a gradient going up here of pH and conductivity. And what that is that you're going along the natural environmental gradient from a low pH, low conductivity in the fresh water up to the estuarine system with a higher pH conductivity. So basically, there's some striking things here. One, the Logan responded to the eutrophication variables, and the other ones are natural estuarine gradient. So from our analysis, we found that these 4, total phosphorus, NOx, and turbidity and pH, were the main variables which were significantly contributed to changes in composition. So it's quite important. But what we really wanted to do was understand how which taxa respond along these variables. So if you can just think about it this way, if you're walking up towards the source of the pollution, say it's a sewage treatment plant, what we're really interested in is that if you hypothetically walk up, on the left you have those taxa that respond negatively to the gradient. As it gets stronger, they start falling out, become less abundant. And also, conversely, you'll have taxa that respond favourably. They'll enjoy the high nutrients, etc., and they will proliferate. And using these measurements here, you can actually get a tipping point for the community to identify where's the largest change in composition occurring. So we're interested in exploring this, and I just want to jump back one slide. It's important to notice that some of these variables are correlated, so you can't really say nitrogen is purely nitrogen, phosphorus is phosphorus, because of the correlation there. But I just want to show you some of the analysis we did just looking at total phosphorus. So you can see here, here's our total phosphorus gradient. So you look along the edge here, and it's running up to about 300 micrograms per litre. And on the left in black are those taxas that responded negatively to an increase in phosphorus. Conversely, on the right are those ones that went positive to phosphorus. And I'm just showing the top 20 of each one here. Basically, for phosphorus, the total phosphorus, we found that about 330 OTUs responded negatively. And 40% of those with diatoms from bacillophaces. And this is a common thread that we find in all our studies, is that diatoms are highly, highly sensitive, good indicators of environmental change. So if you just look here, you can see at about 24 micrograms per litre, there's a sharp drop off of taxa. And this is the confidence in 95% range that they found in, and you can see that those taxa that were responded negatively to total phosphorus were generally found in small ranges and generally fell out here. There's the occasional one that goes against that. That's the most underpinning trend. So conversely, we found that there was 465 taxa that responded positively to total phosphorus and included a large number of diatoms from a different group than bacillophaceae. And what we found is that this happened at around 100 micrograms per litre, and those taxa that responded favourably were found across a huge range of concentrations. So once they started proliferating, they could really handle very high concentrations of total phosphorus. And using this information, we calculated a tipping point that we found that our tipping community maximum change occurred at about 34 micrograms per litre, and the Australian guidelines are about 30 micrograms per litre. But while these two figures are very close to each other, again, I caution the use here. This is based on the time of sampling, and it's an estuary and water moves up and down and total phosphorus concentrations change. And I've done this using model data and you get a similar result. But also, there's the influence of other multiple stresses in the system, so it's hard to get a definitive point. But I think it clearly articulates that you can get a feel from where composition changes, and importantly, identify those taxa which respond both favourably and negatively to changes along environmental gradients. So, in a nutshell, what we found with using metabarcoding, and it's shown in this study here, is that it's cheaper. It's quite cheap to do, especially if you do large batches of samples. It's faster, so it takes us about 4 weeks to produce several hundred samples. It's clearly sensitive to environmental change. And if you use lots of different markers and loci, you have the potential to cover the whole of life, although at various taxonomic resolutions. And also you can use the technology to look at degraded samples doing paleo work and diet research. So it's quite a robust technique. However, there are some negatives with it. And mainly, one, you're relying on PCR, and as we discussed, it has all those artifacts, and that means I don't believe you can get real abundance data out of it. So the abundance of reads really doesn't reflect true abundance of biomass. Even though we use presence-absence data, that still has some issues. So what often we find is that even though if you put a tiger in one sample, it will jump to some of the other samples occasionally. So there's some technical issues associated with that. And you also, if you're using DNA, you're amplifying deceased organisms. Importantly, you still require taxonomic expertise, because if you want to know what the organism is and you want to sequence it properly and get good resolution, you need a taxonomist. And another limitation, it doesn't tell you about function. So for the next part of the talk, I just wanted to talk really closely about another technique we've been using to see if we can sort of attenuate some of these issues. So you can now circumvent PCR by using approaches such as what's called shotgun sequencing, and I call these two different approaches I view it as metagenomics and metatranscriptomics, which is basically, for me, metagenomics is where you use DNA and you get unamplified DNA, you nebulize it into tiny chunks, of 200 or 300 base pairs, get sequenced, and from that you can use a variety of approaches to get taxonomic and phylogenetic information or whole genomes, and you can also use it for functional annotation and look at things like nitrogen pathways, etc., and protein and functional diversity. And then you could use both of these to look at both composition and function. So that's with DNA, and with RNA it's a similar approach, but I refer to this one as metatranscriptomics, and so you can get the same kind of thing. So metatranscript tells you what's happening at the time you sample, and metagenomics sort of gives you more overview across time. So for the next bit of the talk, I just really want to talk about focusing here using shotgun sequencing to look at eukaryote composition. So basically getting that and getting structure out of it. So what we did is we went back to Southeast Queensland, to those estuaries, to 3 of the estuaries we sampled before, which was the Noosa, the Maroochy and the Logan, and we also did the Albert, which is off the Logan, which is the tributary of the sewage treatment plant. And we collected 65 samples and did 65 shotgun sequences. So the first thing we want to do is, so basically get all these little reads, and then we used a approach called KMA, and we use those reads to identify if the fragments from the sequencer could be associated with 16S, which is prokaryotic bacteria, or with 18S, eukaryotes. And so when I did the original analysis, we got some quite funny data. And so digging in a bit deeper, I wanted to see what kind of data do you really pull out of this. Now I just want to emphasize we used 5 samples per lane, so it's quite heavy sequencing. And what we found here, and then what I'm talking about is the number of reads associated, proportion of reads associated with either 16S and 18S. So you can see on the left here is the proportion of reads that we could assign to bacteria, and on the right here is the proportion of reads in red that we could assign to eukaryotes. And I just want to emphasize that this is an order of a magnitude lower than this. So here is our bacterial response, and here is our eukaryotes. So what we found is that if you're just looking at 16S and you get sequence data, we could assign about on average 0.03% of our sequences to 16S. And far worse, for eukaryotes, it's about 0.002 of our reads we can do to eukaryotes. And that's because you've got all that other genetic information in there. So you've got all the genes and everything else in those genomes, and we're just trying to fish out the stuff we're looking for. In comparison to when we did the amplicon, we had a whole vial of the genes we're interested in. So you can see, even using quite heavy sequencing, you're getting a very shallow profile of both 16S and 18S. So what does this really look like from a species richness perspective? Here's the samples I've done, and you can see here in this case looking at eukaryotes, and this is the richness of the samples. And you can see when the black is from the old technique using amplicon sequencing, and the red is using shotgun sequencing. So what we found with shotgun sequencing is that we had about an average of 107 eukaryote OTUs per sample. But when we did with the amplicon, we had about 4 times that with 484. So you get far greater richness from the amplicon sequencing. However, you lose that proportional abundance thing very poorly because of the amplification. So you can see it's growing using shotgun provides you in very complex samples quite a shallow perspective of your biodiversity. And this was somewhat of a disappointment because I thought all my problems had been solved. So what does this really mean from an ecological point of view? So if we just go back to our estuaries again, so here is the Noosa, the good one, the Maroochy, moderate, Logan, the bad one, and I've just got the tributary coming off the Logan here called the Albert. And I've got an ordination plot with the 18S amplicons, that's the old way of doing it, with the shotgun using abundance data, and 18S with presence absence data from the shotgun. And you can see, using all approaches, you can actually discriminate between the different estuaries. So that was fine. However, only on the old approach, we can actually discriminate between the marine interface and the more freshwater components. These sites here are purely marine, so they're right out the front of the estuary, and the rest is starting to get where the conductivity starts to change. And we weren't able to get that discrimination using the shotgun approach. And this is something we consistently find, that we find that using, because of the shallower sequence we're getting with the shotgun, it's not as responsive to environmental variables as the other approach. So when looking at complex systems, I really strongly recommend reliance purely on shotgun data if you're doing composition data. If you look for composition information, I'd strongly suggest doing both approaches. So, in conclusion, I just want to say that I believe metabarcoding is transforming the way we do benthic ecology. It's faster, cheaper, and more sensitive in detecting environmental change. You can look at multiple taxonomic groups. They can be sequenced simultaneously. You still need, if you want species level taxonomic resolution, you still requires a custom database and a taxonomist, which I think is a great thing. However, there's some challenges in interpreting the data, so that's why you've got all those different taxonomic groups and trying to understand what they do and how they play is a huge challenge. I believe that shotgun sequencing is really important because it can provide you functional data. However, I have concerns about its issue when you're looking at composition. So that means we still have a challenge about quantification. And even if you could get pure quantification, sequence everything, I think there's still a number of issues that the sequence reads doesn't actually mean abundance of taxa or biomass. You know, some organisms have lots of genes, copies of the genes. Some organisms are big, some are fat. Some organisms grow more cells as they get bigger. Some of the cells get bigger. So there's huge challenges there. And so I think it's probably best to look at it a different way and not trying to make it abundance and biodiversity per se, but just look at it as reads as a different measurement. I think one of the other great challenges is that there's substantial evidence still required for its adoption into the community. However, in Australia, the resistance is quite subsided. So now metabarcoding is going into the water quality guidelines as a line of evidence. And I think one of the important things is that, you know, like all approaches, you really need to have very good, rigorous and transparent quality control assurance protocols. And I think this is essential for getting to adoption. Also, just don't send your data off, get it back and run with it, because often things aren't working well and you have contamination or the sequencing is not great. And finally, I just want to say it's only one approach for obtaining ecological data. I don't think sequencing data resolves everything. And I think it's important to ensure that we've got true ecologists doing traditional data, because if you look at the slides here, it's more than just a bit of sequence. You've got to understand how an animal behaves with its environment. You've got to understand symbiotic relationships, predator-prey relationships, and behavioural relationships. And without this, it's just a bunch of sequence data. So it's really important to, though it's really important data and it's really good data, it needs to be embedded with other ecological lines of evidence. And thank you very much for your time today.
Great, thanks, Anthony. So we'll move right on. I think there are a lot of questions we can explore from that talk. But as I said, I think we'll move right on to Aina, and then deal with questions for both talks at the end. Aina, the floor is yours.
Thank you, Peter. Thank you, Anthony. I will talk you about some methods that we have developed to estimate, to assess the environmental risk to pollution in harbour areas. So as Peter said, I come from IH Cantabria in the north of Spain. We do a lot of research, education, and also we transfer numerical models and different tools, and we are involved in the different working groups of the World Harbour Project. So as you know, harbours are complex systems. In harbours we have human activities that introduce substances, materials to the environment. These kind of substances affect to the ecosystems, and also we have these port infrastructures that reduce the flushing capacity of the water body. So all these systems are very complex, and we need methods, techniques to provide to the harbour managers in order to prioritize and to know what kind of measures or where they should apply before and then after, where are the hot spots in their harbours. For this reason, we have been developing different methods in order to solve different problems with the environmental risk assessment. So I will show you 4 of our methods in order to estimate environmental risk assessment in different spatial scales, in different levels, different methods, ones that are more simplistic or ones that are more sophisticated because they need numerical models in order to transport these substances and to have a spatial variation of the environmental risk, and other ones that the first one that is just very qualitative for a port manager or regional government that wants to know how are they marinas, are they good, are they producing a lot of impact on the environment, where they should put an eye on in order to apply measures and reduce their impact. So I will first start for the more simplistic one that we have developed for marinas, and then I will go through the other 3 methods. So let's see the method that we have developed for the marina. for marinas at the regional, national, and international scales. We were thinking like some regional manager that wants to know what kind of pressures their marinas are impact on the environment. So that's why we want to provide this method to this kind of stakeholders that they could apply without any support. So because of that, it's fast, it's simple, and it's a low-cost methodology, we could say. So with some of parameters and indicators and some thresholds, they can easily apply these methods in order to know the impacts, the consequences that each of the marinas have on the environment, and also considering some of characteristics of the environment, and also the environmental management, the measures and the systems that are they applying at marina level. So we will see now the method, the indicators, and the parameters that we use to, let's see now. So we have developed a very easy method, considering the risk as the consequence versus vulnerability, and also a parameter, a factor that assess the environmental management of each marina. So regarding consequences, we have select 3 of the main activities that are developed in the marina: navigation, dredging, and the surrounding activities around the marina, having into account the density of boats, and also if the marina has or not gas station or dry docks. We estimate the frequency of dredging considering the marina's topology or the closure depth, and to estimate the consequence of the impact of the activities that are around the marina, we use land uses, the current land cover. Regarding vulnerability, we have into account susceptibility, considering the flushing capacity of the water body in the marina, the ecological value related to the environmental singular elements that are inside the marina or even close to the marina, and also the naturalness regarding to the marina's typology, if it's a harbor, is a dock, is an interior marina, or it's just an anchorage that they don't have any port infrastructures. We also estimate environmental management, considering if they have measures to manage weight, to manage different waste residues in the marina, and also if they have applied environmental management systems like EMAS here in Europe, ISO, Blue Flag, or whatever system that they are applying it. So we estimate the risk, and then we can classify these marinas at national, regional, international level, even between very high and very low risk. So we have applied this method to 320 marinas in Spain, and this way we can have all the environmental risks that those marinas are affecting the water quality in their area. So a stakeholder, manager could know where he could apply the measures that in order to reduce the risk. We have more higher risk values in the Mediterranean because of the lower flushing capacity of these marinas, and because they have lots of berths and lots of pressure in those marinas. So moving on to a more sophisticated method, we developed a method to estimate environmental risk for each contaminant source in a harbor area. So thinking of a harbor manager that wants to know, that has identified and characterized each of the contaminant source produced by their activities or even by other one's activities, they can know by this system which is the risk of each of the contaminant sources, so they can prioritize the measures where they have to apply the measures in order to reduce the risk. So we obtain a single and unique value for each of the contaminant sources. So this method was developed for the Spanish National Port Administration in Spain. So we is at harbor level for the contaminant sources that they identify, and it's also for point contaminant sources, for diffuse contaminant sources. So we estimate the probability of occurrence, the consequences of the contaminant sources, the contaminant source, and then also the vulnerability of the affected aquatic system by this contaminant source. As we did it for the first of the methods, we estimate the risk considering a simple formula, simple expression, and we define different parameters and different indicators to estimate each of the factors. In that case, we use numerical models in order to estimate the extent of the area that is affected by point contaminant sources, and we use geographical information systems to know the area affected by this kind of contaminant sources, because we don't have the flow, we don't have the concentration, so we use another kind of tools. So to estimate probability, we have into account the frequency of the contaminant source. We have also into account the hazardousness, considering what kind of substances and materials are handled and are discharged into the environment. We have into account that, as I said, the extent of the contaminant source, how many area is affected by this contaminant source, the recovery of water bodies, considering the persistence of the discharged substances and materials, and also the social repercussions. The vulnerability is estimated by the susceptibility, and also if we have measures to detect, control, and defend some systems that alert the manager to know if the contaminant source is produced, that they can do something to in order to contain or to control this contaminant source, and also to the operational procedure efficiency level. Once we have estimated the risk for each of the contaminant sources identified at the harbor area, we classify them into 3 categories: high, moderate, and low risk. So for a port manager can start for the ones that have higher risk than the other ones. So in order to reduce the risk and have all the contaminant sources into the low category. We have applied this method to different harbors in Spain, and also, let's see, in the north of Spain, in Santander, and also in Gijón. We applied this method also in Huelva Harbor and in Cádiz, and in the Tarragona in the Mediterranean, and Tenerife in the Canary Island. We are talking about that in each harbor we identify like more or less 30 point contaminant sources and around 20, 30 diffuse contaminant sources. So we estimate the risk, the environmental risk with this method for about 60 contaminant sources. We also calibrate and validate this method in the Atlantic ecoregion. It's been a European Interreg project. We had Huelva also, Aveiro, Algarve, Santander, Cherbourg, Burdeos, and also Falmouth and Belfast, and we had the opportunity to validate and calibrate this method. And also in IH Cantabria, we have developed a decision support system that if you introduce all the information that the methods needs in order to estimate the risk, this system can automatically calculate the risk of each of the contaminant sources. So now we have seen two methods, that one that uses the numerical models in order to know the extent, the area that is affected. One very simplistic that we would like also to have one kind of these methods for harbors, not just for marinas, for big harbors. We are now just with the parameters, the indicators, and the threshold. We would like to apply to different harbors, maybe in the World Harbor Project network, in order to calibrate and validate. So we have seen in this method that we use numerical models, but we at the end just obtain one single value of risk for its contaminant source. But now I'm going to show you two methods that at the end the final product is a map, is a risk map, where we can see the spatial variation of risk. And we have two levels of two spatial scales. At facility level, just for one activity in a harbor, and one for the whole harbor, considering all the contaminant sources that are producing effects, are impacting the environment. So we are going, let's see the facility level method. So this method was developed in a Spanish research project, and having into account that oil handling facility and oil spills are one of the main problems in harbors here in Spain, we thought that we could develop a method that facilitate the development of contingency plans and also the design of monitoring and control programs for the managers of the facility. So in that case, we define a method that has into account different environmental risk scenarios, and then using numerical models, we obtain the spatial variation of the probability to affect the environment. So let's see a little bit of our parameters and also our indicators. In order to define the risk scenarios, we have into account spill types and also metocean conditions. For the spill types, we gathered information of local database incidents. So at facility level, they provide us what kind of pollutant incidents they have had during the last years, and we define some typical spill types. So by just considering the discharge quantity and the product density. On the other hand, we define some metocean conditions that are representative from the study areas. So considering the spill types and also the metocean conditions, we have the risk scenarios that we are going to have into account in order to have this risk map, because each of the risk scenarios is linked to a frequency of occurrence. So each of the risk scenarios are modeled, the oil spill is, the hypothetical oil spill is modeled using numerical models, and after 2 hours we have maps of presence and absences of this oil in the surface, and then we multiply each of the maps by the frequency of occurrences, and then we sum up all of them and finally have a risk map. So here, sorry because it's in Spanish, but here is a summary of the results that we obtain in the Tarragona Harbour applying this method. So in Tarragona Harbour is in the Mediterranean. We have a monobuoy that is here. We collect all the information from the local databases, and we define 12 spill types, considering 3 discharge quantity and 4 density, product density. So we have 12 spill types, and also we have 4 representative metocean conditions. Considering the 12 spill types and 4 representative metocean conditions, we have 48 risk scenarios. So for each of these 48 risk scenarios, we obtain this map of presence of oil, and then we sum up all and obtain finally this risk map for the facility, for the oil handling facility at the Tarragona Harbour. So when we have seen the method at facility level, I would like to finish with one method that we have developed in order to have the risk map for the whole harbour, having into account all the contaminant sources that are introducing substances and materials into the environment. So this instrument allows to establish preventive measures and also locate the economic resources and identify what kind of measures we could implement. And also we can also design the monitoring program in those areas that have higher risk. So in that method, we integrate all the environmental risk from multiple effects from different hazards, from different contaminant sources. We do it at harbour level, and again, we have into account the consequences, the impact of the different contaminant sources, and the vulnerability, the environmental characteristics of the environment. This method was also developed in the Maquarie research project. So here the parameters and the indicators that we have used were we estimated the effects of point contaminant sources, diffuse contaminant sources, and also pollutant incidents. For point contaminant sources, we use numerical models, and having into account different thresholds from the European legislation, we estimate the acute and the chronic effect of the chemical pollutions, eutrophication, and also bacteriological pollution. And then we obtain for each contaminant source extent and affected area, potential affected area by each of the point contaminant source. And considering the hazardness of the materials the handled materials in diffuse contaminant sources and also pollutant incidents, and using geographical information systems, we obtain also the area affected, the potential area affected by each of these diffuse contaminant sources and also the pollutant incidents. We have used different methods in order to combine, to integrate all these effects, multiple effects. We have used average methods, worst case method, and also one that weighted method that considers that the point contaminant sources have more impact, are more important than the other ones. And we have obtained different results that each of these methods can be used with different purpose. And the vulnerability, again, we define, we estimate the vulnerability considering the susceptibility, having into account the susceptibility by estimating the recovery time, that is transport time scale related also to the flushing capacity, but we obtain this spatial variation of recovery time. We consider the ecological value, having into account the environmental singular elements affected by these contaminant sources, and also the naturalness by having into account the hydromorphological pressure and the impact. So once we have estimated the consequences and the vulnerability, we obtain a risk map we obtain risk by each of the grid cells in our study area, and we classify these cells, these grid cells, from null risk to very high risk. So here I show you an example that we apply this method to Tarragona Harbour in the Mediterranean Sea. As you can see, we have lots of point and also diffuse contaminant sources. And here you can see the different results that we obtain from the different integrated methods, the average value method, worst case method, and weighted method. What we see is whatever the method is used, the higher impact is always inside the harbor, because we have more activities, more contaminant sources, and also because we have lower flushing capacity. So that's all. Thank you very much.
Fantastic. Wow, a lot of information in those two presentations. Fantastic. And two, somewhat different but equally critical approaches to trying to understand the health of harbors and estuaries. So, let's open it up to questions right away. So, Karen, we ready to bring everybody else in here?
Yes, I have a question from Stuart Pearson in Canberra first. Stuart has put some questions forward for Anthony. He said, great presentation, good to see the progress. So he has 3 questions which I will try and summarise. The first one being about a publication titled A Rapid Genetic Approach for Assessing Sediment Biodiversity and Function that you co-published in 2010. The question there is, given us information, what can we do in response? The second question is, are these cores available as records through time? And then thirdly, with the fall in cost and the increase in speed of analysis, we should be collecting DNA material more routinely, but is there a simple and reliable way to gather it when doing other bio or geophysical surveys without excessive costs?
Wow, thank you, Sarah.
In 4 words or less.
So my memory's already gone. So the first one, you're talking about a report that was a while ago, and I think that was very old technology. That was microarrays originally. So I think things have moved on quite dramatically from there. So I'm not sure how you'd like me to elaborate that on. So the second one, what was the second question again, Karen? Sorry.
So that was around the cores. Can they be used as a record through time?
Yes, you can. So we've actually got work we're doing at the moment. So what we've been doing is collecting deep cores going down 3 meters, and we go and cut the core and do eye tracks on it, so we can look at how the chemistry has changed over the years and if there's waters come in and there's bushfires. And then we slice that up, and then we do dating on it and do sequencing. So it's a different type of you're only using small fragment approach. So we've started building up the biodiversity going back about 3,000 years. This was done up in northern Australia. So what we found is we can go back 3,000 years, but we can't actually go back 60 years because there's so much energy on the top sediment that it's all turned over. So that was a bit of a shame because we really wanted to see where human impacts were coming in. But I think we're going to continue looking at working into more stable environments. So that whole paleo work is taken off, and I've actually got a new postdoc coming over who works in that area, who works on rivers, lakes, and that. So it's a really exciting area. Third question was about rapidness. And again, I think there's a whole bunch of technologies getting developed at the moment. I know there's some work going on in the States looking at real-time sequences in the water to look at particular things. And we're also looking at, and looking for funding support too for it, is to develop a field approach using the, people might be familiar with the little USB sequencer made by Nano Oxford Paul. So what we're hoping to do is to be able to develop a technique. You can take the samples out, do them in the field real quickly, sequence them that afternoon in your campsite, and do some quick bioinformatics and get a feel for what's happening. So I think definitely there is a whole shift towards that faster thing. But again, the problem with the faster technologies is that throughput's lower, and as I showed in my slide, when your throughput goes down, your version of biodiversity changes. So I think it's a space to watch, and I still think there's a lot of work going into it, but it's a very exciting area, but I think it's going to take a few years before it settles down to get robust data. I think you can get coarse trends, but I'm not sure about robustness at this stage. Hope that answers the question. Feel free to contact me if you've got any more or clarification.
Okay, there's another question from Stuart for Aina, and this goes to some of the social and political systems which are quite important. He refers to...
Karen, just to interrupt, so can we not get Stuart on audio?
I'm not sure if he has a microphone. Let me just check. Stuart, are you there?
Yes, I'm here.
Okay, yeah, microphone.
Turn up your volume, Stuart.
Okay, sorry, I haven't got a very good setup here. I was just sort of being in the dark and enjoying it. You can't see me, can you?
No.
But we can hear you.
Okay, I'll try and put up my my volume. Thanks very much. So good answers, Anthony. I think it's an exciting project, and I've been talking about it with lots of people over the last few years. But the questions, thank you very much for the answers. They've all been answered, I think. I haven't got anything else. My question, though, for Ainia, a great talk, thanks. The social and political system's quite important, and I did see that you'd included that in some of the risk assessment. And I think the legal mechanisms and community are really important, and I've sent a reference to you about one of my students who did some work in Sydney Harbour and also in China, and we discovered that there were huge cultural differences between the risks in harbours and the way that risks can be managed. Do you find differences within Spain, to northern Spain versus southern Spain, perhaps, about who can use risk assessments best?
No, we didn't find any difference between north and south of Spain, and even in the Atlantic region that we had this European project, the method was well accepted. We would say that they like it, they could understood, and they could apply it, and they didn't have. We didn't detect any difference in Europe. Maybe because we have the same kind of European legislation that it's behind this kind of methods, because we use the thresholds from the European legislation. So maybe because of that, because we have this similar framework, that's why we didn't detect this kind of differences.
Yes, that's it. That's a good insight. Thank you very much for your presentation as well.
Thank you.
Okay, we have a question from Federica. Federica, are you online?
Yes, but did you hear me? No.
Yes.
Yes.
Ah, okay. Yeah, we have a question for Anthony.
Yeah.
We would like to know how you did perform the reference database for the metagenome metabarcoding.
So on these ones, we used the online ones, so we used silver on that study. We used silver. So in some of the other work on the freshwater stuff, we use customized databases because there's already lots of benthos have been sampled and already taxonomically identified because they're used routinely. So unfortunately, on these ones we use silver. But I also just build a small one as I'm going. So as I'm collecting polychaetes, because that's my background, I'm quite familiar with them. So I start building those as I'm going and building multiple gene reference databases on the taxa. So I think it works fine for the macro, but you're still not going to get there on the meso and the micro though, because you can't work out what's what isolated sequence is. So Frederica, it's really quite, I think it's quite a challenging thing that. But I think it's really important if you're looking at marine invasives or something like that, then you absolutely have to have a database, a customized database there, I think.
Yeah, customers. Okay.
Can I add a follow-up question? So, Anthony, do you have any comments about the advantages of using of sequencing eukaryotes, you know, 18S essentially, versus doing the bacterial piece. I mean, certainly in terms of some of the relative abundance issues, some of the complications go away. You know, they're all the same size, they're all single cell. There's been some analyses of the extent of, you know, number of gene copies and how that influences things. So do you have any comments about the advantages of, you know, broadly the two kinds of marker genes?
Yeah, you hit the nail on the head. I think 16S technically is an easier gene to work with and to get quantification out of. But then you've got plasticity in bacteria, because I know a dog does what a dog does and a cat does a cat, but we don't have gene transfers, and my cat's acting like a dog. So, but then again, conversely, you can get functional attributes, right? So I think, to be honest, I think it's a holistic approach is warranted. The issue is that when such data falls in the hands of me, I don't know anything about diatoms and chytrids and bacteria. So this is the big challenge, is that there's so many taxonomic groups out there. And one of the ways we're trying to really understand is go past composition and start looking at the way communities are put together using things like network analysis. So it's not purely based on if an organism's present, it's how it interacts with the other organisms. And I think that's the big step forward. And as you're aware, that's a lot of our focus and energy is really going on to explore the commonalities of how systems are put together. You know, you have different systems that have a sample from the Amazon to the desert, but there's characteristics about the way systems put together, symbiosis, predator-prey, and all those, that I think are universal values that might really help us, you know, understand the effects of contaminants. So just from a different perspective, I think.
Mm-hmm. Great, thanks.
So I think that's all for the question. No, I think that's all we have for the question.
No one else waiting?
No, that's all the questions for tonight, and I think we're running a little over time as well. So.
Okay, well, fair enough, I can follow up with Aina separately. So, but let me just thank both of you. You know, I think, as I say, both approaches and both bodies of work are just critical for trying to understand what's going on in harbors. So both very interesting and very illuminating. So thank you, and thanks for all the folks who dialed in or clicked in or whatever they did.
Thank you, Peter.
Thank you, Peter.
Thank you. Cheers.
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 Bye-bye.
Bye.